Okay, hello Marco, I'm Jason Hoeksema, as you know. I'm wondering if you can first introduce yourself, and to that end, it would be great if you could describe your current position and a little bit about where you have previously been, and then just a brief overview of your research interests.
Yeah, sure. First of all, thank you very much for the opportunity to talk a little bit about my work. Yeah, so my name is Marco Cosme. I am currently professor, tenure track for professorship at the Université de Picardie, Jules Verne in France since December 2023. My current focus is now to study the role of the plant microbiome in plant adaptation to climate change. Before coming here to France, I was a postdoc for several years in Belgium, first at the Catholic Université de Louvain, and then later at the University of Antwerp. There I did a great deal of work on the research on mycorrhiza and also on the microbiome and soil microbial communities. Before that, I was a postdoc at Utrecht University in the Netherlands, looking at comparison between compatible and incompatible mycorrhizal plants in their interaction at the molecular level with arbuscomycorrhizal fungi. And before that, I did my doctoral of natural sciences at the Freie Universität Berlin in Germany, where I studied the role of soil biotic interactions on the plant traits that are important for human nutrition.
Okay, super, thank you. So just starting big picture a little bit, how did you become interested in conducting research on mycorrhiza in the first place?
Yeah, so my bachelor was in forestry. So in the beginning, I was interested in forest management, and I didn't start working straight on mycorrhiza research. I first did some work on the forest regeneration, and during that time I became particularly interested in the role of plant biotic interactions in determining the success of plant survival, but also plant growth. And in my particular interest on mycorrhizas came from the fact that these fungal plant associations seems to have such an important role in plant nutrition, and also not only in terms of plant uptake of macronutrients, but also in plant uptake of micronutrients. That at that time I was particularly interested in plant quality as a food source for humans, and so I had a particular interest in knowing how these microbial associations could influence the variability in micronutrients in plants.
Okay. Well, let's switch and talk a little bit specifically about your excellent paper published last year. Mycorrhizas drive the evolution of plant adaptation to drought, which I very much enjoyed reading. It was published in Communications Biology. I was wondering if you could tell us first a bit about the origins of this project. How did you come to the idea to conduct this synthetic research project?
Well, so the topic itself, which is the use of phylogenetic comparative methods, came as a solution in response to the circumstances of the COVID lockdown. So during that time, I was starting a Marie Curie postdoc fellowship, and I had prepared all the resources I needed to perform experiments in the greenhouse. And then we had the lockdown. I lost my plans. And yeah, the Marie Curie was just 2 years, so I had to make a very quick strategic decision in order to have enough data so that I could continue to publish results. So at that time, I was interested in comparing non-mycorrhizal with mycorrhizal plants at the molecular level, and I made the switch to do this type of comparisons at the evolutionary level, because I could harvest data from databases that were already available, so I would not be so much dependent on the wet lab work. So that was my switch of topic. I had to learn a few new methods. But the topic of this particular paper originated, so at that time I learned these new methods, and while learning about these methods, I also proposed to teach a course at the Humboldt University online that was called Ecological Interactions in Climate Change. What can we learn from past evolution about the future? And the students had to perform group work and choose a type of organisms where they can look at how different ecological interactions affect the evolution of traits that are important for adaptation to climate change. And initially I used this topic of drought tolerance as an example, as a case study to teach the methods. But then the results were quite promising and interesting, and then I performed additional analysis, and it started to gain form for a publication.
Okay, that makes sense. I definitely appreciate your approach to utilizing synthetic research to help fill in a gap in your research progress where COVID had created a big challenge. This relates to some advice that I sometimes give to graduate students about planning their overall research career, is that synthetic projects like this, like meta-analysis, can really help to diversify your portfolio of approaches that you use, and that can help to fill in time periods when you are kind of between experiments, between grants, if an experiment goes wrong, or during a period of time when you're not publishing something else, you can always work on this kind of project. And so yeah, that's really smart. I appreciate that.
Yeah, definitely. Especially when you are a postdoc and you have only a 2-year grant.
Yeah, absolutely. That's really a good strategy. Well, so I was wondering if you could summarize just a couple of the key findings from the communications biology paper, speaking to a general audience.
Yeah, so the study focused on testing for correlative evolution between two traits. One is the mycorrhizal strategy, so whether plants form arbuscular mycorrhizal symbiosis. In my paper, I grouped ericoid and ectomycorrhizas together as a state, or when plants don't form any of these associations to obtain resources from the soil. So and the other trait is drought adaptation, that I also simplified in whether a plant is drought sensitive or drought tolerant. So the main finding is first that there is a correlated evolution between these two traits, and what this means is that the speed of evolution of drought adaptation, which is the changes between drought sensitive and drought tolerant, depend on the mycorrhizal strategy that a given lineage has. So that's the first aspect, and vice versa. Also, the changes in mycorrhizal strategy depend on the drought adaptation that the lineage have. So this is the first finding, and the second finding is that the plant lineages that form either ericoid or ectomycorrhizal, they support faster transitions between drought adaptation states, comparing with the lineage that form arbuscular mycorrhizal, or the lineage that do not form any of these types of mycorrhizal associations. These suggest that mycorrhizal associations, they do facilitate the evolutionary process of drought adaptation in land plants.
Mm-hmm. Okay. Yeah, that's really interesting. And specifically, one thing that caught my attention, and correct me if I'm wrong, was that I think looking at the title of the paper, it might be easy to think, oh, mycorrhizae facilitate drought tolerance, but really it's more about this mycorrhizal symbiosis facilitating a higher evolutionary rate back and forth between drought tolerant and drought sensitive strategies. Is that correct?
Yes, that's correct. That was also a surprising result for me because I came from this perspective that mycorrhizal associations promote drought tolerance. So I would be expecting to see faster transitions from drought sensitive to drought tolerant states. But actually what the models detect is that the speed of evolution from drought sensitive to drought tolerant is almost as fast as the speed of evolution from drought sensitive from drought tolerant to drought sensitive. When the plants are hosting ectoericoid mycorrhizas, but also arbuscle mycorrhizas. So it seems like mycorrhizal association, they just have a main role of facilitating the evolutionary processes of changes in the drought adaptation, regardless of whether this is a gain of tolerance or a loss of tolerance.
Mm-hmm. Okay. And do you have an intuitive explanation? I think you discussed this in the paper for why the rate of evolution may be higher in the ecto and ericoid lineages than compared to arbuscular?
Yeah, so this is a working hypothesis that needs still to be tested. What I believe that is happening is because we see a good relation in terms of the diversity of fungi that can associate with ectos, mycorrhizal plants, which is much more larger than the diversity of fungi that can associate with arbuscle mycorrhizal plants. And if we assume that this taxonomic diversity is also related with functional diversity, so the explanation that I propose is that this higher functional diversity might cause an effect of increasing the phenotypic variability in plant populations. And what this means is that in environments where you have a fluctuating natural selection, which is the case of drought, so drought is an unexpected event that happens from time to time. So when you have increase in fluctuating or this type of selection, having what it's called a diversified bed heading strategy increases the chances of these plant populations to have at least some individuals that are able to survive to the next generation and increases the chances of adaptation. And what I believe that is happening is that there is a mycorrhizal mediated diversified bed heading strategy. So this higher diversity of fungal associations in the ectomycorrhizal plants probably increases the phenotypic variation in terms of drought tolerance, and this allows to have always a few individuals in next generation, so it helps to reduce extinction and increases the likelihood of adaptation.
Mm-hmm.
So this is a working hypothesis that I have to explain these different rates of drought adaptation transitions.
Yeah. Okay. That's a really interesting idea. I wanted to ask you, what was the biggest challenge you faced in conducting this project?
Well, from the scratch, I had to learn new methods, so that was quite challenging. My R programming skills were also quite limited, so I had to develop my skills there. But I had good contacts. So the package that I used was published by James Boyko, and which is someone that I collaborate, and they provide me very good lights on how to utilize and interpret the results of this package. And because these processes, they are very computing demanding, so I had to learn how to use high performance computing facilities. So all this was new to me, but it was very interesting to learn it and put it into practice. Then I think another challenge, because I wrote the paper alone, was to try to build a very synthetic story, without having discussions with co-authors. So I had to think from the start to the end what I wanted to put into place, which was quite challenging, but also quite rewarding to arrive at the end with a story that I was satisfied. It's also a good experience because I had more liberties to go in the directions that I wanted to go. And in the end, I was quite satisfied with the result.
Yeah, I had been also planning to ask you about that. Probably that in a lot of these big synthetic research projects, it's pretty common to have many authors on these papers. And so it's a bit unusual to see, single author papers on these, on a single author on such a high profile paper with such a big data set. And so, but also I did want to say, I feel like it's inspiring for junior scientists to see this, because they can see that it can be done, and they don't have to have necessarily a gigantic collaborative network in order to conduct a project like this. So I guess I had been meaning to ask you, what was your experience doing this project, by yourself? You touched on that a little bit already, but I wonder if you have any further thoughts about that.
Yeah, so I think for me, it was a very good experience, and I recommend. I think it's a good way for someone to prove to themselves that they can deliver, like as you said, like a high profile, I don't know if you can say that, but high profile manuscript. So I think it's certainly an experience that helped me to understand that I can work on every details that are required to build a good publication from the start to the end. And also that has some it's a lonely process, I have to say. I also like to collaborate, and it's always nice to have discussions about science with colleagues. So building a paper as a single author has that drawback that all the intellectual process is individual. But I think it's worth it, because then you can really put something that expresses your unique vision on the topic. And at the same time, especially for me at the stage where I was as a postdoc, it's a good way to prove that I have capacity to initiate a research line that can be followed up with my own research or the research by others.
Yeah, absolutely. Okay, the final question I wanted to ask you is if you could just tell the audience a little bit about your plans for research going forward. How does this kind of synthetic investigation fit into, your plans for your research program more broadly? Is this a kind of project you plan to continue working on? And are you doing other kinds of work as well?
So this particular paper, it's a good evidence to show that plant microbe associations can play a very important role in how fast plants can adapt to environmental change. And my way now moving forward, I want to still continue to work with mycorrhizal associations, but I will also expand that to the whole plant microbiome. it's quite easy to work with mycorrhizal associations in this type of synthetic papers because there is large databases on mycorrhizal associations, and that was very helpful. But now I want to test similar hypotheses with looking at other components of the plant microbiome and to understand how taxonomic and functional diversity in the microbiome influence the rate of how plants adapt to drought, but also increasing temperatures and other factors that are associated with environmental change.
Okay, great. Well, I look forward to seeing the papers that emerge from all of that work. And want to thank you for agreeing to this interview. It was a really interesting conversation, and again, a really interesting paper as well. So thanks again.
Yeah, thank you. For me, it was a great opportunity and a privilege. Thank you very much.